one. You have logged your very first data point with the LI6800. So this is important that we have done this. Now, there wasn't a plant in the chamber. We will not be using this data, but that is how we log a data point. And then once go ahead and press the log button again. If you press the log button again, you'll see the same prompt come up. It'll ask you again, what species? See? And so then you could either say, oh, it's still the same species, or you could change it. If you don't need to change it, just press continue.
Yeah, continue, yeah. Oh. Close this, yeah.
Yeah. So now we have logged 2 points. So we are doing well. Now, let's say that we are done. We are done logging our measurements. So then we go back to log files.
Log file.
And we go to close log, which is the big red button at the bottom. Oh, perfect. Now you see 2 things. We had the log data file that we just created, and it says it's 10 kilobytes. And we have a second file here that is logdata.xlsx. That's our Excel file. At the moment, it appears to be 0 bytes. Don't worry about that. It will update correctly. But sometimes it appears as 0 bytes for the first few minutes until the file system scans again. So don't worry about that. It will not download as 0 bytes. So that is how we open a log file, how we create a user constant, how we log a data point, and how we close a log file. So these are very important steps because we would like you to do this on your own. We would like you to go out and take a survey measurement experiment before class tomorrow. And so we need you to know how to do this. The next thing you need to know how to do is how to get the data from your instrument to your computer. And there are 2 ways to do this. The easiest way is to use a USB drive. So if you have a USB drive, what you can do is insert it into one of the 2 USB ports on the side of your instrument. Let me spotlight the, yep, there we go. So the USB port is there. And if you have a USB drive, which I think we have a spare drive, just a moment.
አሁን ያላችሁበት ክፍል ውስጥ አለ እንደ አበባ ነገር or something? ክፍል ውስጥ ለዛሬም ባይሆን ለነገ ማለቴ ነው ግን ክፍል ውስጥም ከሌለ ነገ እስካሁን እንትን ባትሪው ቻርጅ ያደርጋል ውጭ ወጣችሁ መሞከር ትችላላችሁ አሁን እንዳሳየችው ኦፕን ሎግ ብላችሁ ቀጠል ላይ እንትን አርጋችሁ 1-2 measurement ሞክሩት nasty I think
Okay, so you could see that I inserted a USB drive here on my instrument and then let me share my screen in case you don't have one. Let's see, I'm going to share my screen.
እንትን በእንትን ያለ ማለት ነበር የተተከለ እንደዚህ የተቆረጠ ሳይሆን የተተከለ ለነገ
So
yes we have
አሁን USB ተከታተ
Okay.
Okay.
አለን ያው በእኔሚስ በጥሰን ስላመጣ ነው
All right. Let's get back to the downloading data.
a USB drive.
Sorry, wait a moment.
Okay.
Do you have a USB drive? Do we send USB drives? Okay. USB ያለው ሰው አለ እንዴ? ፍላሽ ድራይቭ?
Yes.
እሺ ይክበር። እስቲ አሁን ዳታ ዳውንሎድ አደራረጉን እንድናየው አብረን
Okay.
ከኋላ okay.
I see the USB drive is being inserted. Excellent. Okay. So what we're going to do is we're going down to manage files. I'm going to go down to manage files. Yep, perfect. And then you see that on the right side, we have your USB drive, and it always has a weird letter number combination that doesn't make any sense if you're not a computer. But what we need to see here is that it's there, that it sees the USB drive, that it doesn't think that there is no USB drive. So what we can do is we would like to copy the files to the USB. The first thing we need to do is change directories because we're currently in the home directory on our instrument and we would like to be in the log directory. So if you tap on the blue triangle next to home directory, you can go down to log directory, the third option, yep. And we have two files here, and you see that your Excel file is now showing not as 0 bytes, but 15 kilobytes because the software was able to update. So go ahead and select both of those files. It usually works to kind of tap and pull to select them both. Perfect. Okay. And then you now see that it says copy two files using how much space and which files am I copying. Go ahead and press copy. Finished copying files, press close. And then you can eject the USB drive by pressing the little eject button. And it says safe to remove. That is how we download data from our instrument. That's the easiest way to do this. Go ahead and close what, yeah, okay. So then what you could do is then remove that USB drive from the back of the console, and you can put it into a computer and open the Excel file as you would normally do. You can transfer files by connecting to a computer like a network drive, but honestly, it's a lot easier to use the USB drive because you can just, unless you're doing a large amount of copying. If you need to copy an entire week of data or something like that, then connecting with the network cable often works better. Simply because trying to select all those little files with your fingers just might drive you a little bit crazy. But if you're just copying one or two files, USB drives are much more easy to work with. So that is how you have now opened a data file. You have logged data point, you have closed a file, and you have copied data to your USB drive, and you could then open it on your computer. You have done pretty much everything that you need to do for the assignment that I'm going to give you. So I'm going to share my screen, and I'm going to talk about the assignment first. And then we are going to do a few extra things to get ready for it. So let me share this. Okay, so you have homework. No, that's not what I wanted to do. You have homework. So what we're going to ask you to do is make some survey measurements. And you can see that this is slide 26 of 27, which means that there's a lot of stuff before it, and we will talk about the theory tomorrow. But we want you to have some time taking some measurements on some plants. It's been a lot of staring at the computer and staring at the instrument. So let's actually take some measurements on some plants. So what we'd like you to do is make some survey measurements before class tomorrow. Ideally, if you can, we'd like you to measure 3 species, 3 plants each, or if you have only 3 species, you could do 3 leaves. Or if you only have 1 plant, you could do 1 plant and 3 leaves. You know, we just want you to collect some data. And then if you would like to use fluorometer options to flash on log, we can do that. And we will also configure your instrument to match if. We haven't really talked about match criteria very much, but what we want to do is tell our instrument that it decides when we match. We talked yesterday about what matching is and why matching is important. And in a lot of cases, to make sure that we match when we should, it works well to have the instrument decide if we need to match. That way we don't match when we don't need to, and we do match when we do need to. And then what we're going to do is watch graph A, which is the first graph for stability. And what do I mean by stability? I basically mean that after you clamp the leaf, we would like the line to kind of level off. That tells us that, okay, everything is stable. If it's still going up, then that means that the plant is getting used to the environment that it's in. If it's going down, it doesn't like the environment that it's in. And that doesn't work well. But so here your homework is to make some survey measurements. So you need to open the log file and you need to log the data. And that's one of the reasons that we set up that user constant. And then we're going to do with that. So this is the homework. And now I will stop sharing again so that we can configure your instrument for matching and logging, which we can't do most of the time when the log file is open. Some things we can do with the log file open, some things we want to have the log file closed before we set it up. So what I'd like you to do is go to matching options. Yep. And our options are when logging, never match, always match, only match if. What do we mean? Well, never match is easy. Never match. Always match is also easy. Every time we log, we matched. And that might not be needed. Or we can choose only match if. And only match if says, look at the conditions that the leaf is in and the machine is in and decide for yourself. So let's choose only match if for CO2. And the criteria are only match CO2 if delta CO2 is less than 10 ppm. No, leave it as only match, please. The criteria that we currently have checked are if delta CO2 is less than 10 ppm, or if reference change is more than 100 ppm since last match, or time has elapsed. And remember from our theory lecture yesterday, we talked about some of the matching criteria, and they were if we have a small delta, not much difference between reference and sample, or if CO2 changed a lot since the last time we matched, or if time had gone by And so we want to say, okay, is everything still stable?